Students, good morning to you. Today we will be discussing about phloem. What is phloem? It's a conducting tissue. It serves as a channel. It helps in transporting food from the leaves to the underground parts. And this phloem is a complex tissue. That means it is made up of more than one type of cells. Even though they are made up of more than one type of cells, they all work together as a unit, and they consist of the following elements, or they consist of the following type of cells. Number one is called sieve elements. Number two is called companion cells. Number three is the phloem fibers. And fourth one is phloem parenchyma. Now, what are sieve elements? Sieve elements are conducting elements of phloem. They serve as a channel and they are collectively called as sieve elements. So the conducting elements of phloem are collectively called as sieve elements. So they help in transporting, or they help in guiding, or they help in leading. What they lead or what they guide or what they transport through them, the food materials. Now these sieve tubes are elongated. They are very lengthy cells, or lengthened, stretched very long. These are very long cells. And these cells are tube-like, just like tubes, slender, very thin, and they are placed end to end. So these sieve elements, these cells are placed one above the other as a vertical row of cells. And in these cells, the transverse walls, that is, the horizontal walls, are perforated. Perforated means they have pores or openings. That is why we call them as sieve. Sieve means perforated openings. So you can find small, minute pores. So the transverse walls at the ends are perforated. That means they have small openings, and that is why they are called as sieve plates. Now, what is the main function of the sieve tube? The main function of the sieve tube is translocation of food. That is, movement of food from one place to another, from the leaves to the storage organs of the plant. So leaves, they prepare food, and they are carried by these phloem, and they are stored in the underground roots, underground stems. For example, underground roots or tap roots, for example, carrot, radish, and beetroot. Likewise, they are stored in the underground stems. For example, tuber, that is potato, then corn, colocasia example, and bulb example is onion, and rhizome example ginger. So these are all underground stems where the food is stored, and this food is prepared by the leaves, and they pass through the phloem. That is, phloem carry these food to these underground parts. And the next cell is called companion cell or companion cells. What are these companion cells? These are closely associated with the sieve elements. So these cells are very closely associated or attached to the lateral wall of the sieve tubes. Lateral, sidewards. So they are attached to the sides of the sieve tubes. And these are elongated cells. These are also stretched very long or lengthy cells, lengthened cells. And these companion cell may be equal in length. So when you compare the companion cell and the sieve elements, that is sieve tubes, they are of equal in length. Sometimes they are equal in length, or sometimes the mother cell, that is the companion mother cell, it may be split or may be divided into a series of companion cells, a row of companion cells. So it may be split into series of companion cells, a row of cells. So these companion cells, they are giving, what do they give? They give mechanical strength to the sieve tube, because through the sieve tube the food is passed. So when the food is passed in a solution form, it has to withstand the stress of the physical forces. So the companion cells, they assist, they help the sieve tubes to carry the food from the leaves to the underground storage organs. Then you come across the third one is called phloem fibers. What are phloem fibers? Phloem fibers are nothing but they are Sclerenchyma cells associated with primary and secondary phloem. So when the sclerenchyma cells, we studied already, isn't it? We studied about two types of sclerenchyma cells. One is called fibers, and another one is called sclerids. What are fibers? They are elongated cells with pointed ends. So when the sclerenchyma cells are associated with the primary phloem and secondary phloem. What is primary phloem? If the phloem is formed from procambium. Procambium is a meristematic cell. You know, its duty or its function is to divide and produce new cells. So if the phloem is formed from the procambium, and that is called primary phloem, and secondary phloem is nothing but it is formed from the vascular cambium. Vascular cambium is formed in between the xylem and phloem. So if the vascular cambium gives rise to the phloem, then that is called secondary phloem. For example, you would have heard the word bark, the term bark. What is bark? It's a term applied to all the tissues outside the vascular cambium of the stem. All the tissues means it includes the periderm, the cortex, the primary phloem, and the secondary phloem. So all these are collectively called as bark. So this is sclerenchyma cells, that is, these fibers are associated with primary and secondary phloem, and these are also elongated, lengthened or lengthy cells. Lignified. What is lignified? It comes from the word lignin. Lignin is an organic polymer, and that is very useful in the formation of cell walls, especially in this sclerenchyma, xylem and phloem. These cell walls, they contain more lignin, and they lend rigidity. They lend rigidity. That means they give mechanical support. They are very stiff, rigid, and also they do not rot easily. They will not decay so easily. No rotting. And also they provide mechanical strength to the plant body. They give ability to withstand the stress of physical forces. So they give mechanical support to the plant body. And the last one is phloem parenchyma. Phloem parenchyma, what are they? Parenchyma associated with phloem is called phloem parenchyma, and they are living cells. Why they are called living cells? Because they have nucleus, they have cytoplasm and nucleus. What is their function? They store food materials. So phloem parenchyma, they store food materials. So these are the four different types of phloem cells or phloem elements. Now we move on to the differences between xylem and phloem. Xylem, it conducts water and minerals. It transports water and minerals. Phloem, it conducts organic solutes, that is food materials. Food materials, they flow through the phloem just like the blood flowing through our blood vessels. So food always, it flows in a solution form. Then number two, conduction is mostly unidirectional, that is on one direction only. From the roots, water is taken up to the apical parts of the plant. Apical parts means stem and leaves. But phloem, the conduction may be bidirectional on both the sides or both the directions. First, from the leaves they prepare food and they are sent downwards to the storage organs. And then from the storage organs, food is transported upwards to the growing parts of the plant. And the third one, conducting channels or tracheids and vessels. So channels means passage, a pathway, passageway. So here in xylem, it is tracheids and vessels. They only conduct or transport the water and mineral salts. But in phloem, the conducting passageway or sieve tubes, they transport the food. And what are the components of xylem? The components of xylem are xylem tracheids, xylem vessels, xylem parenchyma, and xylem fibers. In this, the xylem parenchyma is a living tissue. It's a living tissue because it has the nucleus and cytoplasm. And what are the components of the phloem? The components of the phloem are sieve elements. In sieve elements, there are two types. You'll be studying in detail in +1 when you come to +1. Sieve cells and sieve tubes. And then comes companion cells. Then comes phloem parenchyma and phloem fibers. Then we move on to the differences between the meristematic tissue and permanent tissue. We studied about meristems, isn't it? Meristem, I told you in the very first class, meristems are dividing cells. They divide, they redivide, and they produce new cells. They produce new cells. I told you meristem is derived from the Greek word called meristos, which means divisible. So its only function is to divide and produce new cells. And we studied three different types of meristems: apical meristems, intercalary meristem, and lateral meristem. Then what are permanent tissue? Permanent tissues are nothing but where the cells have stopped division. The dividing or dividing nature of those cells are stopped either completely or for time being. So when they stop dividing and they switch over to do certain functions, those cells we call them as permanent tissues. So what are the differences between these meristematic tissue and permanent tissue? When you look into the meristematic tissue, the cell wall is very thin and elastic in nature, flexible. But permanent tissue, they are very rigid. Cell wall is thick and rigid. What are the component cells? How are the component cells in meristematic tissues? The component cells are small. They are spherical in shape. Sometimes they are polygonal, many-sided, and they are undifferentiated. They are not differentiated into parenchyma, collenchyma, sclerenchyma, or xylem and phloem. No differentiation. These cells, they remain as such, and their only function is to divide and produce new cells. How about the component cells of the permanent tissue? They are very large, and they are differentiated into parenchyma, arenchyma, chlorenchyma, collenchyma, and sclerenchyma, fibers, sclerids, xylem, phloem. So here they are differentiated, and they have different shapes. Some are spherical, some are polygonal, some are round in shape, some are elongated in shape. So they have different shapes. Then in meristematic tissue, the cytoplasm is thick, dense, and the vacuoles are nearly absent. What are vacuoles? Vacuoles are organelles. They are found in the plant cell, and they consist of cell sap. So vacuoles are nothing but membrane-bound organelles filled with cell sap. That cell sap consists of salts and sugars dissolved in water. So vacuoles are nearly absent in meristematic tissue, but there in permanent tissue, there is a big central vacuole present. And in meristematic tissue, no intercellular spaces. These cells are very compactly arranged. In between these cells, you don't find any space. But in permanent tissues, like parenchyma, like collenchyma, you find in between those cells lot of spaces. And those spaces are called intercellular spaces. And in meristematic tissue, the nucleus is very large and prominent, noticeable. You can see the nucleus very clearly in the microscope. But in permanent tissue, the nucleus is less conspicuous, So the nucleus here in meristematic tissue is large and prominent, noticeable. You can see it clearly under the microscope. Even the prominent, the other word for prominent, is conspicuous, clearly seen. But in permanent tissue, the nucleus is less conspicuous. Conspicuous means less prominent. You cannot see it very clearly. And in meristematic tissue, the cells grow and divide regularly. So their only function is to divide and redivide and to produce new numerous cells. Permanent tissue, these cells, they don't divide. They do not normally divide. And meristematic tissue, they provide mechanical support and elasticity to the plant body, flexibility. Even if it is stretched, it is not broken. And permanent tissue, they provide only mechanical support. That is, it helps the plant to withstand the stress of the physical forces. So with this, we complete the phloem tissue, the differences between xylem and phloem, and also the differences between the meristematic and permanent tissue. And we will continue in the next video. Thank you very much.